Cells use chemical signals that they release into their environment that can be sensed by other cells of a similar type. And my lab in particular is interested in a specific group of these types of sensing protein molecules called G proteins. And G proteins are particularly interesting to us because they have great prominence and importance in human health. So it turns out that baker's yeast have G proteins. And they use these G proteins in the same fashion. The G proteins are a very similar structure to what we have in our own human cells. And we use this model system in particular to understand how G proteins function after they become altered by a natural chemical process known as post-translational modification. And post-translational modification is a process whereby very small molecules can become attached to a protein, like a G protein, and change the way that protein behaves and functions. It might change where the protein is located inside the cell. It might change the catalytic activity of the protein if it's an enzyme. It might change who that protein interacts with. And so we're particularly interested in understanding how post-translational modifications, these small chemical attachments, can alter the way cells communicate with one another. And so a major aspect of my research program is to use a very powerful technique for looking at these very, very small modifications. It's called mass spectrometry. And we use a particular type of mass spectrometry commonly referred to as protein mass spectrometry. So we look at very small changes in the mass of G proteins after they've been modified in different ways through these post-translational modifications.